Those 7 papers I'm going to be talking about have addressed every part of this damage. Specifically, paper number 6 over there, published in 2010, showed that Protandim prevents that loss of this antioxidant enzyme, PON1. Number 6 and number 7 showed that Protandim prevents this damage to lipids, to polyunsaturated fatty acids in our body that give rise to T-bars. Papers number 3 and 5 showed that Protandim prevents damage to DNA. In fact, those papers showed that Protandim supplementation could prevent skin cancers in the mice that were used in that study. Papers 4 and 6 showed that Protandim can prevent fibrosis, scar tissue that can cause a heart to go into failure. Paper number 4 showed that Protandim can prevent apoptosis, or programmed cell death. And you'll notice that in many of these things, multiple papers have addressed the same issue. And there's method to that. I think some people look at the specific disease models that have been studied in these papers, and it may look like a random selection. Why are we looking at drug-induced pulmonary hypertension? Why are we looking at intimal hyperplasia in veins that are used to bypass clogged arteries? It's because it's these same 5 processes that are involved in free radical damage, oxidative stress-induced damage throughout the body. What protects us from oxidative stress is not one antioxidant enzyme or even 2, even though that's what we focused on in paper number 1. It's an entire system of antioxidant enzymes. And here we see a tiny corner of that system. The enzymes are shown in blue on this sequence of reactions. The bad products are shown in red, and the detoxified products, or the antioxidant products that help get rid of the bad stuff, is shown in green. It's the blue enzymes that make these reactions occur. The first paper really focused only on that little part. It looked at the reactions catalyzed by acetylene catalase. A second paper, which is one of the very important ones, showed that the enzymes involved in making glutathione, you hear a lot about glutathione in this industry, are also upregulated by Protandim, allowing our bodies, our cells, our individual cells to make more glutathione, not because the precursors or the raw materials to make glutathione are limiting. Sometimes they are. More often, it's the enzymes. It's the things encoded by your genes, upregulated by Protandim, that allow you to make more of these things. Protandim contains 5 phytochemicals. They come from 5 different plants. All of these plants have a long, long history in traditional medicine. People didn't know how they worked, but for centuries they knew they do work. What they didn't know was that they are regulators of a protein called NRF2 and activators of NRF2. And that's the mechanism whereby more than 500 of your most protective genes are regulated by these simple, believe it or not, chemicals produced by humble little plants that have been recognized in India and China for centuries and centuries. They activate what's called the master regulator of stress-related responses of antioxidant enzymes. How does Protandim activate NRF2? How does Protandim activate NRF2 in a cell? This is a cartoon that illustrates that. This purple shape in the upper right corner of this slide represents one of those 5 chemicals from plants, from medicinal plants. And so as Protandim approaches a cell, the cell actually has receptors on its surface. And my analogy here is this is like if the cell is your house, Protandim just stepped onto your front porch. And it rings your doorbell when that happens. And that causes a change inside your house. You hear the chimes or the bell. And you know something has just happened that may change events within your house. In this case, an enzyme on the inside of that cellular house called a kinase, has been activated, signified by turning yellow. Well, the business of that kinase is to put phosphate groups, to chemically modify a handful of molecules that it knows how to modify. One of those is that little red oval labeled NRF2. NRF2 is being held in the cytosol, outside the nucleus where the DNA resides, by this other larger protein called KEAP1. So the activated kinase now chemically modifies NRF2, and you can see that little yellow P that's been added. That's a phosphate group. So it's chemically changed NRF2. And the protein that's holding on to NRF2 now sees this thing has changed its shape. It doesn't fit my grasp quite as well. It releases that NRF2. So it's now free to diffuse around the cell, and it's small enough it can actually enter the nucleus, which is the vault. It's where all the plans, the blueprints for everything you are reside in the form of DNA. So this activated NRF2 now has access to all of those blueprints, and it binds to specific genes within there. Out of 25,000, at least 500 or 600 bind activated NRF2, and the result is those genes are turned on. Each of those genes has a switch, like a dimmer switch on the lights in your house. NRF2 simply turns that knob. Those genes express what they encode. The products designated by the blueprint of each and every of those genes is now produced. And among those products are antioxidant enzymes, anti-inflammatory enzymes, antifibrotic enzymes, all kinds of stress-related genes that help cells, especially when they're in chemical stress, when they're in oxidative stress. It helps bring things back into balance and to provide a normalizing influence on those cells. We can visualize this microscopically. The upper three panels show control cells, cells without Protandim. We can stain NRF2, and it stains red in this slide. It's a fluorescent red stain for NRF2. We can also stain the nuclei of those cells. It's staining the DNA. And if you merge those two, you get what you see in the upper right. What you see, if you look carefully at that, is the nuclei are green, and the NRF2 is red surrounding the nuclei. It's not where the DNA is. If we treat those same cells with Protandim and now stain the NRF2 and stain the DNA in the nucleus and now superimpose the red stain and the green stain, what you see is the lower right figure. Everything looks yellow, and that's because if you add red light and green light together, what your eye sees is yellow. This proves to scientists looking through this microscope that Protandim treatment caused the nerve to go from its location outside the nucleus to exactly where the DNA is. That is, the NRF2 has moved into the nucleus, and that's where it can regulate those genes, turn up all of those processes. The first paper published in 2006, I've condensed into one or two sentences for you here. This is really what you don't need to know about everything in that paper, but you do need to know this. After 30 days of supplementation, T-bars, which is oxidized lipids, declined by an average of 40%, and the age-dependent increase was eliminated. By 120 days, SOD and catalase had increased by 30%, 54% in the red cells, respectively. That's very significant. This shows what T-bars looks like in a population of varying age. There's a lot of scatter. We're not all the same at age 40 or 50 or 60. Some of us are in great shape. Some of us are already suffering from diseases associated with aging. If you draw a linear regression line, the line through those scattered dots, you can see that there's a relentless progression as you go from 20 to 80. Your T-bars averages go up and up and up. If you look at people in this group who were taking conventional so-called antioxidant supplements that many of us took for decades.